Today, we're going under the microscope. We're going to follow the clues, hidden deep inside human tissue, to see exactly how a pathologist unravels the story of a cancer cell and makes a diagnosis that changes everything.
So let's just start with one number that really frames this whole conversation. 60. A habitual heavy smoker has a 60 times, that's 60 times the risk of developing lung cancer compared to someone who doesn't smoke. It's a pretty staggering figure, and it really underscores why we need to understand this disease inside and out.
So, how do we get from a huge global statistic all the way down to a diagnosis for one person? Well, we're going to follow the exact path a pathologist takes. We'll start with a wide view, the crime scene, then we'll identify our primary suspects, and then we're going to zoom all the way in to find the microscopic fingerprints that absolutely seal the case. But before we even step into the lab, we have to understand why this work is so incredibly important. Let's just take a moment to look at the sheer scale of lung cancer's impact around the world. Yeah, this is not a minor issue. It is the single leading cause of cancer death on the entire planet. And it's a disease that usually shows up between the ages of 40 and 70, striking people right in the middle of their lives. So what's causing it? Yeah, things like industrial exposure to nasty stuff like asbestos, air pollution, and even some genetic changes. They all play a role. But one cause stands way, way above all the others: tobacco smoking. It is, without a doubt, the single most important driver here. Okay, so we're moving from the cause to the effect. When a pathologist in a lab gets a lung sample, they aren't just looking for cancer in general. No, they're hunting for a very specific culprit, one of just a few primary suspects. So you can think of lung carcinomas as being broken down into these four major types. Think of them like the four most wanted criminals in the world of lung cancer. Each one has a different backstory, a different look, and, crucially, requires a different battle plan. Now, each of those four types has its own unique story. But for today, we're focusing our investigation on just one of them: squamous cell carcinoma. So what exactly is it? Well, it's a major player, and it has an incredibly strong link to smoking. So strong, in fact, that people often just call it a smoker's cancer. And for whatever reason, we see it statistically more often in men. But what's really fascinating is what's going on at the molecular level. This isn't just random chaos. It's often driven by very specific genetic mistakes. For example, think of a gene called TP53 as the brakes on cell growth. Well, in this cancer, those brakes are often cut, which gives the cells a permanent green light to just multiply out of control. And this uncontrolled growth, it doesn't just happen overnight. It's more like a gradual takeover, a step-by-step progression from a healthy cell to something truly invasive. Let's trace that path. It all starts with something called dysplasia, which are these early precancerous changes. From there, it can progress to what's called carcinoma in situ, or CIS. Now, at this stage, you have real cancer cells, but, and this is so important, they haven't broken through their original layer of tissue. They're contained. It is a critical window for detection. So we get the what and we get the how it develops. Now, for the most important part. How does a pathologist actually see this? What are the clues they're actually looking for? Well, it's really a two-step process. First, the pathologist looks at what they can see with just their naked eye, what's called the gross morphology. But then they move to the incredible detail of the microscope, and that's where the final verdict is really found. So with just the naked eye, the tumor can show up in a few different ways. It might grow inward, kind of like a blockage in a pipe, or it might grow outward, literally breaking through the wall of the airway. Or sometimes it just forms this distinct cauliflower-like mass right in the lung tissue itself. And when the pathologist actually slices into the tumor, its physical appearance gives them the next set of clues. It's usually this gray-white color and feels firm. And in bigger tumors, you might even see areas where the tumor has literally outgrown its own blood supply and started to die from the inside out. All right, let's move to the microscope. This is where the definitive evidence is found. This is where a suspicion finally becomes a diagnosis. Under high magnification, the difference is just stark. Healthy tissue is orderly. This is total chaos. You see these disorganized nests of cells. Their nuclei, the brain of the cell, are all different shapes and sizes, and they stain really darkly. And the main body of the cell, the cytoplasm, that usually shows up as this vibrant pink. But Among all these different clues, there are two features that are the absolute smoking guns. When a pathologist sees these, they can say with certainty, this is squamous cell carcinoma.
The first is something called keratinization. Now, keratin is that tough protein that makes up our skin and hair. These cancer cells, it's like they remember their origins as protective squamous cells, and they start producing it. Sometimes they make so much of it that it forms these beautiful concentric little swirls called keratin pearls, which are just unmistakable under the microscope. The second key feature is these tiny intercellular bridges. They're these little spiny connections that physically link one cancer cell to another. They look exactly like little grappling hooks, just holding the whole tumor mass tightly together.
And this is the whole point, right? Identifying these specific features isn't just some academic exercise. The patient's entire outlook, their whole treatment plan, it all depends on this exact diagnosis, on how advanced the cancer is when it's found, and on just how abnormal those cells look under the microscope. And that's what's so powerful here. Every single detail, from the color of the tumor to the presence of those tiny intercellular bridges, it all contributes to a diagnosis. It's a story that's literally written in the language of cells, and it's that critical first chapter that determines everything, and I mean everything that follows for the patient.